Did I go to your personal page to grab the link to share?
Hang on.
Okay.
All right. We're going to go. We're live. All right, people, ladies and gentlemen, Dr. James Lyons-Weiler coming to you from the WWDNYK studios in Pittsburgh, Pennsylvania. And if you haven't recognized the voice of my guest so far, you certainly will in a moment. He's unmistakably the Wild Doc. How you doing, Dale?
Doing well, James.
Good, good. My
Doing
friends
real
call me
well.
Jack, so he must be pissed off at me. What's going on?
Oh man, I always go back and forth. I'm like, Jack James, because I've got it like on my, it's Jack in my phone on text messaging, but then it's James here on Facebook.
I apologize for the microaggression, but let's dispense with all cultural propriety here. This is a fantastically crazy time. And we're very fortunate to have people like you who, instantly can recognize when you read studies, and you read a ton of studies, that there's something wrong when there's a report that there is nothing wrong. There's a mismatch between what's being said in the study, the conclusions of a scientific study, and in the press releases about a study. And we've seen that elsewhere in other aspects of public health and in medicine, for sure. But
Yep.
you stumbled into a fairly egregiously obvious nail to hit with your hammer. So what did you find?
So, ultimately, there's a level of curiosity when I read these research studies. I'm always asking, why did they ask the questions that they asked? Why did they test the cytokines that they tested? Immediately, my thought was, okay, if this mRNA vaccine is capable of producing a single protein, basically through genetic modification, to simplify the explanation, it's a genetic modification, stimulating our own cells to produce these proteins, these S-spike proteins. So it enables us to identify this virus easier. So that's what the game plan and goal is of a vaccine. If it's a protein that's not similar to our own body tissues, then that molecular mimicry that you've been able to really kind of, whittle out in the science and research and genetic sequencing of these proteins. I love your work there. Well, then you may not have that, the prior animal studies, having antibody-dependent enhancement of disease process. You may not have that because there's different proteins that I think there was one single protein that didn't have it. I had to go back to my references to look at it, that didn't have that effect. But a lot of the animal studies in mice were seeing enhanced disease pathology. The virus
and you covered that at length on your podcast, which is where you actually helped me bring that to me. What's your podcast again? Where do you go live?
they'd have to go back to, I don't know the exact titles, but on the wilddoc.com. Of course, if I may, I've been in for you as well. If you've got email campaign and stuff because of censoring right now, other people being censored. I'm starting to get more and more of my videos taken down, and I'm like, I literally read directly from NIH research study, right, and YouTube says it's dangerous. I'm like, so reading research in the public arena is dangerous. But so yeah, I've got several out there, and I did talk briefly on it. I think I've sporadically talked about it in the past, but I know that, I talked about the fact that they've been doing coronavirus vaccine research and development for 10 plus years now.
Right.
So, and they're different type of vaccines in the past. They're not these mRNA, the Moderna's mRNA vaccine.
I pinged you over to have a look over one of the studies because there was a missing monkey. One of the sets of the monkeys in the Moderna study. It's supposed to be 8 in a group, and they ended up with 7. And I dug into it and found that it was a sedation problem with the monkey. And just like every other outcome in vaccines, the presumption is there's just something wrong with the monkey, not that the vaccine made the monkey intolerant of sedation. So here I am. I'm thinking study design. I'm thinking are they sufficiently powered? Do they have enough animals? What, at that level, and then you come at me with this micro missile, this tiny little detail that nobody in their right mind would ever see in a study. You nailed it. Where you saw that there was a disruption, a change in the expression of a very important protein.
When I was looking at so you got to look at what they measured, and then you're basically, they are saying that because they didn't see substantial Th2 immune responses, their kind of qualifying factor for the idea that the vaccine was safe and wouldn't cause what they were referring to in their study. It was almost close to VAER. It wasn't VAERS, that abbreviation, but it was
VAER. It was the vaccine adverse. VAER
enhanced disease syndrome or something, I think. Yeah,
it's respiratory disease. So vaccine enhanced respiratory disease. That's what it stands for. But so let's back up a little bit. The study is touting as, wait a minute, this thing might be a really good thing because we don't see an overly Th2 response. And the Th2 response, many people know, are associated with prone to autoimmune conditions, prone to chronic illness, prone to pro-inflammatory, right? Prone to cytokine storm kind of outcomes, not just with COVID, but in general, with general health. And so they're saying that this is potentially a good thing. And then that you wrote to me and you said, hey, wait a minute, there's something here with this IL-21. I need to take a look into it. You're pretty excited about it, actually. So,
well, I mean, just taking and reading through, and I think you've got the image there now, is the Journal of Autoimmunity, Clinical Significance and Immunobiology of IL-21 in Autoimmunity. You read that one single paper, and that was only published in 2019. Reading more on IL-21, they only discovered IL-21 in 2000. So this is new information. Mm-hmm. And it's only been the last few years that they've been able to really kind of start to weed out what IL-21 does. And it is one of the most pro-inflammatory. It promotes the development of autoimmune antibodies, self-antibodies. It actually downregulates T regulatory cells, which are, there's studies out there now on Kawasaki disease, multi-organ system inflammatory syndromes, all the things they're saying COVID causes. Well, the vaccine is going to cause what they're determining to be a causal factor in immune dysregulation that leads to these syndromes, that leads to these problems. Mm-hmm. And so like I was saying, it's like, why did they choose to measure these biomarkers? To me, it was interesting that they said, like, okay, we didn't see here's their exact quote from the monkey study on this vaccine, was that CD4 type 2 helper cells, the type 2 immune responses. They were saying that interleukins 4, 5, and 13. Well, they only measured 4 and 13. They didn't even measure 5. I'm like, so if you said 5 has a bearing on this, why didn't you measure it? Yeah. It's just, I always have suspicion when I'm letting the fox guard the hen house. And especially with vaccine manufacturers that have already gotten millions of dollars and they're going to get billions and billions if they put out this vaccine. They're going to get trillions in nationwide or worldwide. So they measured certain responses, and you look at interleukin 2. It's all I can say is it's kind of like read that article on the clinical significance of and immunobiology of IL-21 in autoimmunity. If this happens to be something that will promote maybe 50%, this was 8 weeks. When they did this monkey study, it was 8 weeks after these injections, right, these vaccines, that they tested their antibody levels. So, now I'm like, okay, so yeah, you did some tissue samples, you looked at the monkey's post, you are these IL interleukin 21 that can cause severe autoimmune conditions that can exacerbate autoimmunity, that they're literally developing. AstraZeneca is developing drugs in the autoimmune market for these Janus kinase, but that's the name of it, but to inhibit IL-21.
Oh my gosh, you're kidding me. I was going to say, I didn't want to interrupt. I was going to say, this time next year, we're going to hear about IL-21 inhibitors being on the market. So that's a riot,
No,
man.
James, this is what I was saying. Like, this is why I got so excited, because it's like I'm like sitting back thinking to myself, like, seriously, God, is this for real? I did a video yesterday on my Facebook page, and I got the links there. But if you go to Market Watch, you'll see tyrosine kinase inhibitors. You'll see AstraZeneca owning one of the companies making the tyrosine kinase inhibitor, which is one pathway to inhibit it. The other one is the Janus pathway, JAK pathway. And it's Janus pathway inhibitors. They were literally, Market Watch was saying on the 27th of July, the day before they published this monkey study
Yeah.
that shows IL-21 is enhanced, not short term. 8 weeks is not short term. We should not have prolonged IL-21 8 weeks. To me,
that's exactly right. We're looking at they said that the respiratory, they only looked for adverse enhancement, disease enhancement, respiratory. It could be associated with IL-5, but they didn't test this. You said that. And then you noted that IL-21s were substantially elevated, and you asked how long will this last. And then you also said, listen, IL-21 is a key role in the development of T helper T17 cells, which contributes to pathogenesis and a range of inflammatory diseases. I'm reading your message. IL-21 promotes a range of autoimmune diseases, including lupus, type 1 diabetes, multiple sclerosis, inflammatory bowel disease, and psoriasis. And you sent a link to interleukin 21, a double-edged sword with therapeutic potential. So that's a nature paper. So that's why you're hyped about IL-21.
I think there's papers on encephalitis. There's papers on Kawasaki disease. Like, it's literally like I don't want to call it a treasure trove, but it is a linchpin. If this causes long-standing immune dysregulation of upregulation or increased IL-21, we are looking at a catastrophic event in terms of autoimmunity. It will take us to we're already at pandemic proportions of autoimmune disease. It's a leading cause of death in young women. I mean, so if you want to talk about years lost, if we were going to say and argue for or against a vaccine,
right,
if this vaccine causes substantial or even a small 10, 20, 30, it was in the microdose, the 10 microgram dose, that was 50% had elevated IL-21. It was 100% in massive increases in the 100 microgram dose.
So IL-21 is, if you have any viral infection or a bacterial infection, we have an innate immune response. It's part and parcel to that. When I see Th2, sometimes Th17, I'm going to think we're looking at a pro-inflammatory immune system that's going to be certainly biased towards the development of autoimmunity if a person is genetically predisposed. We're looking under the hood now at and getting it down to IL-21. It makes you wonder how many of these other conditions. What does the MMR vaccine do to IL-21? What does pertussis, Tdap, and DTaP do to IL-21? It really makes you wonder what aluminum does to IL-21, right?
I mean, I think they've been using the tetanus toxoid to induce autoimmunity before they even knew what IL-21 was. So the pathway using vaccine ingredients to cause autoimmune conditions, it's probably not even understood. They just know it does it. And it's like, kind of like, how did they find out that the tetanus toxoid they inject through a vaccine causes autoimmune conditions? But they use it, but then they claim that the vaccine doesn't cause it because, usually. But you said something there, like genetically predisposed. Well, IL-21 has an impact on genetic expression.
Yeah.
So, like, the impact of this, IL-21
is a regulator of other genes, you mean, right?
It needs to go up to help. It improves viral clearance. So for your immune system to respond appropriately to efficiently clear off a virus, IL-21 has a critical role in maintaining lower levels of HIV, chronic and Epstein-Barr virus, things like that. So it's like you need it, but you want to clear the virus efficiently, quickly, and not have sustained IL-21, because the sustained level of IL-21, it's like you need it to fight a virus, but you then, if it's up and it stays up, then it is causing T regulatory cell deficiencies. If you have that, your Tregs are necessary for you to be able to recognize yourself from your non-self. It is there to say, these antibodies against my own tissues, thyroid disease, Hashimoto's. Look at the prevalence of Hashimoto's. If we increase IL-21, which then increases the cell's efficiency to damage and attack those tissues. So it's going to enhance disease pathogenicity. It's going to create more damage to your thyroid, folks. To simplify it, I don't want to talk science.
No, we need to talk science. We're not breaking science. People that come here, that's why I don't have thousands and thousands of people that watch my show, because I don't break it down all the time. But we can break this one down. Let's talk about interleukins, what they are. They're signaling molecules, otherwise known as cytokines. They perform a variety of functions. The cytokines were first seen to be expressed in white blood cells. They provide a variety of functions that include things like cellular differentiation, the regulation of the immune system. So they're natural proteins that exist in the body. And IL-6, for instance, we know is a linchpin in terms of the brain inflammation process that leads to autoimmunity against brain proteins. Seems to be important in autism. And now, outside of the brain, outside of the central nervous system, IL-21 appears to be, from your research into it, looking at the publications, it seems to be an important linchpin in the biochemical pathway of self versus non-self, potential and modulating immune responses, most importantly. So the fact that we see IL-21 itself isn't an issue. The fact that we have prolonged IL-21 points to, COVID-19, as bad as it is with respect to how messed up society is now, as bad as it is with respect to people that have lost loved ones and got seriously sick. COVID-19 is teaching us a shit ton about a lot of things. Not about our systems. Yeah. It has stressed our systems. It has stressed our society. It's stressed our politics. Now this IL-21, is it normal after you get an infection that you see 8 weeks in a monkey of IL-21?
What I'm saying is it's sustained levels. When I look at other ones, it should go up for a period of time.
Other ILs, you mean? Other ILs?
Yeah, along with other interleukins, IL-21 should go up during acute infectious disease. It should then decline fairly quickly. It's not going, because if you look at it, it is something that helps you fight the virus off, clear it. Once the virus is cleared, it should not remain elevated. It should decrease quickly. So it's and so most infectious diseases do not take 8 weeks to fight off, okay? Again, certain chronic viral infections, which cause a lot of other problems and increase your risk of autoimmune antibodies, they increase your risk like Epstein-Barr virus and others. Yeah. It could be that you can't clear this virus. So there's other pathways. This is a signaling molecule. It's a communication molecule. So it helps your body do what it's telling your body to do, what it needs to do during that time period. Once you've destroyed the enemy, it's time to repair. Now you need to remove any abnormal antibody that was generated during that infection. So your T regulatory cells need to go back up where they were inhibited by IL-21. Now they need to go back up once IL-21 goes back to basically zero level. That's what I would say to people is, if you just look at the baboon study, you look on, and I'll even go to the page of it. If you go to page 6 of that study, if you look at the baboons that were given PBS, okay, those levels of those memory CD4 T cells, and Th2 responses, Th1 responses, CD40L, THF interleukin 21 levels.
Yeah.
Those levels of those baboons not vaccinated is where you should be, Now, there are memory T cells where we want to have capable of responding efficiently and quickly. And that's what we see with what they're showing now is that hidden immunity with COVID, people that were years, even blood samples years prior to this year.
Right.
Okay.
Right.
So if COVID-19 hasn't been circulating, there's other coronaviruses that give us what's called heterosymptotic immunity, a immunity partially to the new virus. So people's CD8 T cells, their T cell responses, their memory T cells, CD4 T cell memory, they are able to produce antibodies rapidly, quickly, be able to fight it off. Okay? But you want a regulated amount of those antibodies. Extreme levels of antibodies can be, well, is the antibodies causing the problem, the pathogenicity, the severe disease, or is the inefficiency of the body to clear the virus causing more and more antibodies to build up in the system? That's arguable one way or the other. But we do know that if we are sitting here, we need those things to go back down. I think you have to see the body as intelligent. And if the majority of people have antibodies go up against COVID-19 and then decline almost to zero, and then it's maintained under the memory T cells to be able to mount the immune response in the future,
yeah,
that is what the body needs, because antibodies are proteins, they're flags. Your immune system gobbles those up, or when they're attached to anything those flags are attached to, the body interprets as being something it needs to destroy, to kill. If it's a cell, it needs to kill that cell. If it's a virus, it needs to remove that virus. It needs to deactivate, it needs to gobble up and remove that virus.
All right. What I want to do is I want to go into the biological processes here. Thank you for all that of what this IL-21 does, and then ask the $1,000,000 question, right? So it's responsible for positive regulation of the inflammatory response, positive regulation of cytokine production, cellular maturation, positive regulation of B cells, positive regulation of tissue remodeling, positive regulation of T cells, immune response, cell proliferation, interleukin-17 production. These are all positive regulations of natural killer cells mediated cytotoxicity, signal transduction, positive regulation of interferon gamma biosynthetic process, tyrosine phosphorylation of the STAT protein, positive regulation of tyrosine phosphorylation, I just said that, and regulation of receptor activity. This thing is absolutely a linchpin in terms of the body's initial immune response. The $1,000,000 question, there's no doubt it's important. It's expressed in so many tissues. It's intracellular, extracellular. It's found everywhere. It's a signaling molecule. The question is, what is a normal duration of elevated IL-21 in response to a viral infection, response to a vaccine? And is it something special about the COVID-19 proteins that the Moderna vaccine is encoding? Now, I looked at all of the proteins in the study that you mentioned, right, with pathogenic priming, and a huge percentage, and this is why I published it. A huge percentage of the targets, the putative autoimmune targets, like a third of them that are targeted are immune system cells, like B cells. And let's see, I've got the list here. B cells are definitely targeted. So that's why we're seeing all this stuff about T cells. Well, not definitely targeted. I can't say that. There's a high probability of cross-reactivity. I should be accurate in what I say. But there's a large number of important immune cell proteins that have very high probability of molecular mimicry and potential for autoimmunity. And so if this thing, let me, for the sake of argument, say this thing came from bats. Let's say it came from bats. Ultimately, it's a virus that evolved in another species that has, a diverse immune system from ours. It's different from ours. And depending on which type of bat it is, it might have a cytokine innate type immune response, and some bats don't. So I don't know about the bat that this allegedly came from, of its type of immune system. But if this particular virus is well adapted to an animal that does not have a strong initial innate immune response, then a lot of our illness might be happening when you have somebody that's Th2 switched, right? They're already Th2 skewed, so they can't exhibit a normal, and I'm going to call it juvenile human innate immune response. I say it's juvenile because kids don't have adaptive immunity to as many coronaviruses and viruses as adults do. They just haven't been around as long. And so their response seems to be a little bit more of a healthy innate immune response to the virus. So now in terms of this vaccine, you go to a rhesus macaque. Well, there's a killer result that's coming out. I forget which journal it's coming out in by Yehuda Schoenfeld and Darsha Kanduk. The rhesus macaque, out of all the primates, all the mammals that could be used to model, has the fewest epitopes, immunoreactive epitopes that match COVID-19 for some reason. It's the worst model. That's coming out. She's asked me to popularize that, and I'll put a link when that gets published. So if we're trying to understand human immune responses to this, we have to look through the filter of, wait a minute, should they have used a different kind of monkey? Should they have used a ferret? And so if we're sitting on a bomb here of a vaccine that might be unleashed, that certainly wasn't tested, or the results of pathogenic priming, immune response, disease enhancement wasn't published. If they have the data, they haven't published that. Yeah. We're sitting on a time bomb in terms of its effect on our immune system, potentially through autoimmunity to our own proteins that would be missed if they went to a macaque anyway. We're sitting on a time bomb in which, even in spite of that, the innate immune response seems to be elevated for a long period of time in a primate, which is not a bat. Then we're flying blind here. Yep. This is not arguing from ignorance. Clearly, the two of us have done our homework on this. We're flying blind. We're walking into the dark in terms of the phase 2-3 clinical trials.
I mean, what I would say to simplify what you just said is the fact that if we have molecular mimicry, and it's not being seen simply because of the selection of the specific type of monkey.
Right.
If we do, then we don't see molecular mimicry in the monkey because it doesn't have similar epitopes, but then we do have it in human populations. We're going to have molecular mimicry, which increases the risk and rate of development of autoimmune against those tissues that are mimicked by the proteins on the virus. Then you have an increase in IL-21. If that transfers over into humans as well, and the sustained level of IL-21 and the downregulation of the regulatory cells to protect against the development of autoimmunity, you've got a huge double whammy there. You've got basically what science is now. I mean, when you read this article, it's like they're basically saying that if IL-1 is sustained, it is literally like, again, like you said, it's a linchpin. It is literally the gatekeeper to autoimmunity. Even just IL-21 alone by itself will enhance the tissue damage in people who already have any autoimmune condition practically. I mean, the study mentions a lot of different tissues: lupus, rheumatoid arthritis, type 1 diabetes, skin disorders. But also, when you look at it, there's the Kawasaki disease. If it enhances Th17 cells, well, that's your autistic children or adults. And then you look at your encephalitis, increased brain inflammation, and so we're playing with fire if this is not a very, very short-term effect that this vaccine would have. But 8 weeks to me is not a short-term effect.
If we understand in humans the clinical course of the disease itself it's like a 2-week disease, right? If you get it, you're going to be sick, and then you're going to have this, unless you're on the Brownstein protocol, in which case you bounce back, right? You're going to be sick, you're going to have fatigue, you're going to wonder, did I, it's like mono. I've had people calling me saying, their family or relative has it, and they're describing the symptoms to me, and they're like, it's like they got mono, they're just wiped out, and then when they feel better, it takes a long time unless they do some interventions to boost the immune system. But if we're looking at it from the perspective of 2 weeks illness, then I still think it's a question. It's an answerable question. How long should the innate immune system be activated following a virus infection? So that's what it boils down to me. That's what science is about. Science is about finding the right and asking the right questions. We could maybe find an answer in the literature, but if it's unusually long in this study, then we still have the filter of, but it's a macaque, not a human, and that's what's so frustrating about vaccine development. How do we ethically test this without challenging people with the virus after they're vaccinated? How do that would be unethical, and yet they're actually talking about doing that. They used to actually do that. So if you look at the coronavirus literature from the 1950s and 60s, when they would challenge humans without a qualm after vaccinating them, they had waning antibodies after 6 months or so. It wasn't like a prolonged, lifelong antibody production. But they had the memory. They still were able to mount a new immune response 12 to 18 months later, after the infection, right? This is after the infection. And so this wasn't after a vaccine. This was after the infection. So in terms of long-term immunity, you're still, if you had a cold last year, you're still able to produce, cold virus last year, you're still able to produce antibodies to the next cold virus.
Yep.
But where do we go if Moderna, FDA, everybody signs off on this vaccine and says in 3 weeks' time everybody can get the vaccine because, of course you're going to see an innate immune response. That's the first thing you're going to see. And they don't finesse the details.
Well, where do we go if they approve this vaccine? I'm going straight to the stock market and going to be investing in Janase kinase inhibitors.
That's a funny comment, but I was going to play devil's advocate, and I was going to say, what if, hypothetically, the IL-21 inhibitors turn out to be a drug that reduces the adverse events from vaccines? What if that happens?
Say that again.
This is me playing devil's advocate, and I know you're a religious man. But the point is, okay, what if the IL-21 inhibitors that are under development turn out to be drugs that reduce adverse events from vaccines? Right now, they tell you to take Tylenol. We know that makes that worse. But what if you can calm down this inflammatory process and allow the adaptive immune system to get its memory acquisition, to learn the antigen source, and calm the rest of the shit down? What if there's a silver lining in all this crap,
So you can use IL-21 to help with cancer patients, they're showing. Metastatic cancer, you can upregulate the massive inflammatory immune response that's necessary to destroy tumor cells.
Sounds just like Coley's toxins. Just like Coley's toxins, right?
Yeah, I would agree with that.
Right.
But when you look at this, if these inhibitors, I think they use some antibodies. So I'm trying to look at this, and I'll try and read directly from this other article. It's not one of the ones I sent you.
Right.
But what they saw was, so they, in certain patients with, I think it's a genetic condition. It's autosomal dominant hyper IgE syndrome. So you're going to have more allergy issues with IgE syndrome. So they have decreased IgG and decreased IgA. So there's a mucosal, and that's a lower non-histamine, non-allergenic type immune response. But these patients not only able to differentiate IgG or IgA producing cells after IL-21, but when they were treated, what they found was they eventually had the body develop antibodies to the drug. And then if you decrease IL-21, you have substantial increases in viral illnesses, especially respiratory illnesses. So if we treat these people, well, now with the way it's like we've just got to stop screwing things up. Like that's my biggest bitch.
It's like
Stop screwing things up.
it's like an oil well. It's like an oil derrick that's burst, and we put the, we try to jam it closed, and then we grab a cow and we try to put it down, and it just can't stop. Mother Nature will always have another trick up her sleeve.
Yeah.
But
So it's like you're going to look if we have to use inhibitors, well, or if they use antibodies and they train the immune system to basically attack IL-21 and neutralize it, well, then you got cancer risk going up.
Then you've got I just read all of the natural functions of IL-21. We don't want to shut it down unless it's out of control. So this would have to be a short duration, short acting drug like Tylenol. Tylenol comes and goes, right?
Let me read you something. From that article I sent you, the one about IL-1 and immunobiology and autoimmunity.
Yeah.
This is something else that was like, to me, I literally sat back and I'm like, this is hilarious, but awesome. Mm-hmm. So let me ask you, what is the herb being used in places? I believe it's Madagascar, certain areas in Africa, and it's being touted as one of it's called COVID organics. What is the active herbal ingredient in that they consider to be one of the most anti-COVID coronavirus herbs? Do you know?
No.
It's artemisinin, sweet wormwood.
Oh yeah, I did hear about wormwood. Yeah, artemisinin.
Yep. So this is Journal of Autoimmunity, 2019. Let me read you directly here. SM 934, an analog of artemisinin. That is a synthetic analog of the active, basically one of the compounds in sweet wormwood, which is artemisinin, with potent immunosuppressive in an autoimmune therapy model. See, wormwood has an antimicrobial effect, antiviral effect. It has an immunomodulatory effect, not necessarily an immunosuppressive effect. But in an autoimmune model, they found that artemisinin, sweet wormwood, impedes IL-21 signaling in a pathway that reduces autoimmune signaling in autoimmune disease. So it's almost like if we were to harness this information, do you see the coincidence there? I'm like, so the herb being used in some countries to help with COVID, and they're literally saying, and I believe it's Madagascar, they're literally saying it's a cure. Like they're like, everybody's just drinking it. We have made this tea. We're selling it to other countries now, and nobody wants to talk about it. Like their president or whoever it is basically got on the news and was like, locally, and was like, we got a cure. If this was a European nation and not an African nation, then it would be up in they would be like, hey, we got a cure, we got it taken care of, no big deal. But he's like, you're basically discriminating against us because we're an African nation. You don't want to use something that's cheap and inexpensive and we created. And they're basically toting low mortality rates, curing it with something. And guess what? The effects of artemisinin and the effects of wormwood are similar to those. They're anti-malarial. It's one of the best anti-malarial herbs out there. Well, what is HCQ? It's an anti-malarial. It's an anti-parasitic. So it's like the drug that Trump and everybody is saying may be a cure in early stages, especially because of, and we could talk pathways there, but maybe not today. But it's like, then we have this herb, and that's where my religious belief is that God provided the answer we need. We just need to use it. We just need to look to him, what he's created, and start harnessing it. But people don't want it. We live in a medicine system, drug model system that ignores all the research on honeysuckle, elderberry, wormwood, nettle, quercetin, EGCGs.
Can't patent it. Oh, exactly. Everybody knows it. Everybody knows that, right? So let's look at this. Therapy. Let's look at this COVID-19 study from Nature. It's coming up here, Dale. Risk for cytokines. Did you see this one? Risk for cytokine targeting in chronic inflammatory diseases. This one's a good one because it actually lists all the cytokines and their activity. It does say IL-6 is upregulated with infection of the virus, and that potentially your JACE inhibitors are good candidates So it's basically an advertisement for that line of research, the Janus kinase inhibitors, the JAK inhibitors. But IL-21's right there, so it affects several cytokines involved in antiviral responses. So these IL-2, IL-15, IL-21, and interferon gamma, those are normal viral responses, and I don't want to get anybody's hopes up, but doesn't that mean that, at least in terms of seeing that, they're seeing what should happen during a viral infection? They don't see the Th2 skew. They see the viral infection-like response. I guess the million-dollar question is it prolonged or is it not prolonged? And that's a great question. So I thank you for raising that. Let's revisit that time period in terms of other studies, because there's got to be a shit ton of studies on the Janus kinase inhibitors.
And it's so Janus kinase inhibitors. It seems to me, just looking at this kind of like drug production, and the article that I shared yesterday on my live was Janus kinase JAK inhibitors market analysis, market size, trend, scope, challenges in pharmaceuticals, biotechnology, and life sciences. Pharmaceutical sector will reach 18.5% in 2023. So 3 years from now, the market size is going to be 18% of the market. 20% of the market will be this specific inhibitor. So it's like, how do they know that it's going to go that route? One, they know that they know that. How's it going to explode? Why do they know that it's going? How do they know it's going to explode? It's just, to me, and that was published May 20th of 2020. So in May this year, they started saying, like, hey, this Janus kinase inhibitor is going to be a top selling drug by 2023. How many years does it take once you elevate IL-21 to initiate tissue damage enough to where people become symptomatic and seek out drug regimens to treat their autoimmune conditions caused by their 2020, October of 2020, or their November of 2020, or their early 2021 COVID vaccine? Just kind of throwing that out there.
No, that's what science is for. You're a good man to ask those questions. I wish if anybody has any idea on what a normal duration of an IL-21 upregulation would be.
Looking for that. And I cannot find it, other than what they see is basically it should be basically close to zero in the normal healthy population. But in the population of individuals with autoimmune conditions, it's elevated. So, and those are elevated levels in those monkeys. So it's,
well, let me say this. Let me say this. One of the questions that you asked that didn't get answered was, why did they bother to measure this? And now we know why. They're looking for what is a normal viral response, right? So the same reason that they looked for Th2 and they said, look, it looks like it's more Th1. And now they're looking at the cytokines and they're saying, oh, good, we're seeing IL-21. The ink in the pudding, however, could possibly be the duration, and whether or not there is any sufficient immunity. These are still open questions. Is there sufficient immunity caused by the vaccine to make it worth society's while to bother with vaccine production, distribution, development, boosters, and everything else? Or are we walking into, like children into the dark, where we say, yeah, look at us, we know how to vaccinate. We vaccinate 100 million people, and then next year coronavirus swings by again, and they get really, really sick. That's the pathogenic priming that I published on. And this initial reaction to a vaccine doesn't tell us about that. So it doesn't tell us anything about whether the degree or the rate of autoimmunity or chronic inflammation conditions that could result from either being vaccinated or getting the virus once or twice. I think the immune system is better at handling something that it hasn't been adjuvanted to be immune to. I think the immune system is well adapted. There are very few viruses that on double infection cause autoimmunity, and if it does, it's probably because you have a molecular a mutation or a change in a protein that makes your protein a lot more like that pathogen's protein. So I'm thinking if this miRNA, which people are afraid of because it feels like a form of gene therapy, people are worried about insertional mutagenesis because you're injecting a nucleotide into the cell. I asked Teresa Deischer, like the second week that first I heard about mRNA vaccines, I called her. I said, Dr. Teresa Deischer, what's the likelihood of insertional mutagenesis of an RNA vaccine from an RNA vaccine? She said it should be no different than the baseline from the virus. The difference then, Dale, is if you're going to vaccinate everybody, you're going to find all the insertional mutagenesis that you can have. Whereas if you have a common cold that comes through or a coronavirus that comes through, we know how to keep the immune system healthy, we know how to minimize the symptoms, that's going to sweep through the population, maybe infect 16% of the people. It's not going to infect 100% of the people. And that's the difference. And people don't get this when it comes to vaccine risk. Well, the vaccine risk is low. No, it's not. You have to look at the population as a unit. If you're going to vaccinate, you're looking at the population level risk, not the individual risk. And I'm not getting upset. I'm just passionate about injecting people's point of view, in their point of view, a different perspective of infection versus injection. With an injection, you could get every single person with laws and mandates, and you could make everybody sick that's going to be sick, whether they were going to be sick with the virus or not. But with a virus, you might get 20% of coverage, so to speak, and you're going to have fewer people sick, right?
Yeah. What I tell people is you can't argue with 100 people who get the virus and get sick from the virus, and you say, well, 5 of them had severe illness and died. Well, in this virus, it's like one of them, maybe
a tenth of one.
It's not even that. Like you'd have to have 1,000. Now one of them got severely sick.
Exactly. That's exactly right.
So the severe outcomes. Mm-hmm. And so if you had that rate in like math simplicity, I'm like, okay, so 1 out of 100. But then what if but only 20% of the population get sick?
Exactly.
And get the virus. But then when you inject 100% of the population,
that's
and
right.
What if it's 1 in every 200? Well, now you have you have 4 score more.
Instead of 1 in 1,000. Instead of 1 in 1,000, it's 1 in 200. And you have to use a booster, so you've doubled your chances, right?
Huge increases, especially when you're affecting the entire population by everybody willfully, some willfully, and some of them maybe by force if they went that direction with this vaccine. Right. Are getting something that has potential to cause illness. And it's going to cause a percentage. And that percentage of the population negatively affected by the vaccine would be greater because of just the fact that it's 100% are being injected. Even if the percentage out of 100 or less injured by the vaccine. And that's just a theoretical argument.
That's right.
But the reality is, what if we're going to get into a situation like we do with the flu vaccine where it causes original antigenic sin, it causes dysregulation in memory T cells and a reduction in CD8 T cell responses to future viral illnesses. It causes, in some situations, flu vaccines have been shown to cause not all of them. So you got to understand that, are they going to have to change the coronavirus vaccine every year? Are we going to get into a situation where this vaccine doesn't have the pathogenic priming, it doesn't have the antibody-dependent enhancement of disease, but the next coronavirus vaccine will be causing that, and will hit and miss occasionally having that. Right. And then what if this one causes the original antigenic sin, where now everybody who gets it before they've ever come exposed to a natural source of coronavirus infection have decreases in their capability of fighting off infection. So now we take where we had periodically throughout history, we may have an increase or a mutation of a virus that, again, arguing whether this is a natural mutation or not, whole different ballpark there. But the reality is we are going to have some pathogens that become more virulent periodically throughout history. And it comes down to the ability for our body to appropriately respond to that. And that is improved and enhanced and improved through many different processes, talking about immunological alone, and our prior exposures can actually dictate how we appropriately respond to this new virus.
Yep.
Now we screw it all up with a vaccine. We don't get that positive feedback within our immunological responses to be able to fight off a more pathogenic virus in the future. Now we go from, they estimated 2 million Americans dying from coronavirus. Now we're seeing, we can argue whether it's true or not, tens of thousands to 100,000, and 3 years, 4 years from now, because of this vaccines and its impact on our immunological responses, heterocytic immunity, all these things like the flu vaccine, and we see constantly increasing levels of people dying from the common cold virus, which is a coronavirus. We take a non-severe virus and we make it severe because the vaccine causes distortion in the immune response.
Absolutely. So the one potential way out of this mess, of course, is that we reach herd immunity first through transmission. And that all depends on what the herd immunity threshold is for the virus, for SARS-CoV-2, right? So we don't know what the real herd immunity is. If you use the mathematical function that is used sometimes, it could be 50, 60%. But the University of Nottingham, did you see that study of the University of Nottingham that the different groups of society have different rates of transmission, and when they accommodate for that, because there's different levels of social activity, that the herd immunity is plausible at 43%. And then there's also the possibility that if in a population that has response efforts to curb COVID-19, that might bring, ironically, the herd immunity threshold down to 10%, because those who are going to get it in a context where there is not a lockdown, but control factors like in Portugal. So
here's an interesting thought I've had about the control efforts. What if we needed to spike quickly, and that's why coronavirus infections year after year before this really were seen primarily, late October, November, and then fell to basically zero throughout all summer months. We're seeing sustained infection transmission now. It may be because of different virus, but what if it's specifically because of our attempts to reduce transmission, so it's slowly spreading, and anytime we come out, we didn't have a spike, we reached herd immunity quickly, enough percentage, and then it eliminated the spread. Right. And then it's going to mutate. We're going to face another one next year, different. It's going to mutate. It's already mutated. You know what I mean? Like, it's not exactly the same virus that started in the original person in China, or wherever it started. Don't even, I just hate people almost like, how much racism is coming about because of this right here? You know what I mean? This, towards Chinese.
We just absolutely don't need that. Doesn't help at all to do that. You're exactly right to bring that up. So if
we're going to have to be,
why does the UK, Dale? I got a question. Why does the UK? Why does the UK, back to herd immunity, say that they may have already achieved herd immunity? Is it because they want to not have egg on their face over the policy, where they tried to wait for herd immunity initially? But what they're saying is that the cross-reactivity from past coronaviruses that circulated through the UK apparently confers immunity. If you look at this objectively, there's no way that probably all of us have not been exposed to coronavirus by now.
We have.
Right. COVID-19, we probably all have, and those of us that are going to get sick have probably already gotten seriously sick, or those who are, that's a form of evidence, right? If you can't identify on your hand, as scary as they made it sound, 5 people that got seriously ill from coronavirus, you're not in a special population, because that explains the experience of every other American in the country. We don't know anybody, all right? The nurse that lives next door to me, she says at the place that she works at, they just forget about coronavirus. It's just another infectious agent. We're going to handle it like everything else. No big deal. That's what the largest medical organizations in Western Pennsylvania are telling their employees. So there's a lot of politicking going on. There's a lot of bullshit going on. I'm really grateful for you for bringing forward this aspect of the science. I love that we ended with unanswered questions because that's what science is all about. I hope it inspires people to go help you. People, please go help Dale find out how long a natural IL-21 response is in response. But I think we're there, man. I think we're at the point where, what added value is there of the disruption of further social distancing and keeping kids out of school? And sure, if you don't want to shake hands, don't shake hands. You want to wear a mask, but let's not enforce each other as if there's a dire pandemic going on when the numbers don't even support a pandemic anymore. I finally got to do the math. In the United States of America, to have an epidemic, you have to have 7% or more of deaths from all causes caused by the pathogenic agent. Yep. Sometime in May, we dropped below 7%, if you look at the percentage of all causes of death. Sometime in May, we got out of the epidemic zone. There's no emergency in the United States anymore. You heard it here on WWDNYK Studios. You heard it here first. I don't know who else has said it. There's too many people are talking out there. But everybody's realizing this could be something that we are, okay, we're almost done with it. Wouldn't that be nice? Right?
Yep.
That'd be nice. So I want to do some more homework with you, Dale. I'll give you the closeout here.
The thought in my brain right now is, what if something like dengue, what if something like that pathogenic priming that is affected and caused by, we would have a natural immune response when we are exposed through our mucosal membranes. That's another whole rabbit hole. But if a large portion of the population has already been exposed to the virus, they've already fought it off, there's absolutely zero need when you have memory T cells. There's no antibodies, but they want to generate antibodies. That's how they said the vaccine's going to work. There's no need for a vaccine for those people who've already been exposed.
Right.
You can't argue there is need, okay?
That's right.
What if there's going to be enhancement of disease process and inflammation through the immunological manner by the vaccine, especially in people who've already been naturally exposed?
Exactly.
If we aren't doing those types of testing, and then we're going to vaccinate a population where a large portion has already contracted the illness, and if there's any type of, instead of the vaccine causing the antibody-dependent enhancement of disease and pathology, it's the vaccine after the exposure. It's the vaccine after the natural exposure. But again, through natural exposure, your mucosal membrane oftentimes has a neutralizing effect. It has a priming effect that's more under control. And like the way I say, if you inject something under your skin, it's directly in the bloodstream. Immediately, there's a different immunological response that takes place than if something comes through the front door. You know what I mean? I walk in my house, the bad guy's already in there. The way I'm going to resolve that situation is going to be a whole lot different than if I hear them pounding on the front door. You know what I mean?
Yeah, sure.
I can have a controlled environment, and I can sit back with my, my shotgun, my rifle, or my pistol, and I can say, you leave the house right now. I'm going to neutralize you really quickly. You come through that door, I will kill you. And they run off because my immune response was at the front door. They didn't get through my barrier.
It's a localized response, exactly. It's a localized response. If you inject it and it's designed to prolong the exposure in different tissue than our basically mucous membranes, epithelial, dendritic cell responses, you're exactly right. So here's what I want to leave people with a concept of hope. We have before us an uncertain future. We have before us people that are acting out of self-preservation, right? We should never, ever again have to shut down society because they have overreached so badly this time. They've pissed off people that are willing to put tens of thousands, hundreds of thousands if they have to, tech-abled people of their own money to put together a tracking database because the government's not doing it. That's the world we live in now. They created that world with their failed testing, right? Their failed contact tracing, with their lies to the public about masks, all of it. They've created a very skeptical society. The people who want data, they want information. So the questions that you're asking, Dale, how do we know that if the people that already had coronavirus, and we know the number of cases that we've had, How do we know that if they get vaccinated, they're not going to become sick? And the government's certainly not going to find that out. They're not going to ask that question. But the population of citizen scientists can ask that question. And so I would be more than happy to entertain contact from anybody that would like to build a database of vaccine adverse events, tracking system, coronavirus adverse events, just to find out who in the population is seriously at most risk. Can we create a prediction model? You're in this population, you're blood type A, you're old, you've got diabetes, you're at high risk. Okay, you need to tell your doctor you need a Th2, Th1 scan and see what your immune system looks like. If it's balanced, good, relax. If it's skewed to Th2, hey, buddy, you got to be careful. And you need to take care of yourself. You need to get yourself healthy, and this is how you get your immune system back on track. That's what I'd love to do. I'd love to take control. I want to wrest control of the responsibility of public health from this centralized bullshit system that is profit-driven and captured, and put it back in the hands of the people. So if anybody wants to help IPAC build that database, I'll be more than happy to man it and put a person on it, help with the adverse, and what we'll do is we'll embed data analysis capabilities right inside of it. So we'll be able to do all the log odds and odds ratios and everything else on your specified populations. Dale, you're a gentlemanly scholar. I know you spend thousands of hours every month reading the most esoteric scientific crap, right? So I thank you on behalf of the listenership and the viewers for bringing you the best that you have. And it's a great question. So keep it up.
I love to get to do it, and I love to do it for the people, and I love doing it because I feel God's called me to do it. And like I said, to be able to find those little things, whether it be the market watch and the investments, this is where to put your money because we're going to need these drugs in the near future, to just that the fact that they mentioned sweet wormwood and artemisinin and and its effect on that, I'm like I use that in autoimmune cases, and I'm like, wow, now I'm understanding more of why it's working. I might say give people hope. If they come to your door and you're not one who's going to put up a physical fight to refuse to get vaccinated, or you're a military member and you're going to be forced vaccinated to maintain your career, please get some sweet wormwood to be taken that while you're getting
after a period of time. After a period of time.
Yeah.
And that's the question. How long before you counteract? All right, man. It's great talking with you. All right. Love you too. Take it easy, bro.